Hi, I'm Laura Hambrick, and I will be talking about the ERBB2 receptors, important beta 1, and transport. So, in experiment 1, we are talking about RNA interference, and we were given this figure, and we had to figure out what the objective was and what this is actually showing us. So the objective that I came up with was analyzing the levels of important beta 1 and ERBB-2 when subjected to nonsense siRNAs, which is expressed in the NS, and it's an expression of important beta 1 reduced, which is the IMPs on the figure, and not subjected to siRNAs, which is your negative, which is also your control. Then to see the presence of the ERBB-2 and important beta 1 in each column. The siRNA and the immunoblotting are great to use with antibodies as it helps siRNA and important beta 1 relationship levels. So by looking at the top panel of important B1, you can see that it shows up in the cytoplasmic and in the nuclear. It does show up in the expression of the important beta 1 reduced. It's just a very faint line, so you know that the expression of the important beta 1 in the nucleus, the reduced, it does work. And the bottom panel, you see that there's the ERB-2. By looking at this, you can see that the ERBB2 has a very high presence in the cytoplasmic fraction right here on the bottom, and that it also has a presence in the nuclear side. But you can see in the expression of the important B1 reduced, you can see that the line for that is not as dark as the other two lines in the nuclear fraction of the ERBB-2. So this just means that important B1 and this go hand in hand. So experiment 2 was talking about the cells expressing either wild type ERBB-2 or mutant ERBB-2 delta NLS. So the objective was the lysates of cells that express either the wild type or the mutant ERBB-2 that is lacking its nuclear localization signal. Then these two types are immunoprecipitated with antibodies to the ERBB-2 or with control MLGG. Then using the immunoblotting to examine the antibodies. Immunoprecipitation is used to separate proteins that are bound to a specific antibody from the rest of the sample. Immunoblotting is also known as Western blotting, and it is performed by solubilizing a mixture of proteins and then separating the proteins by size using an SDS page. So the top panel of this figure is showing that the control antibody MLGG did not have a presence in either the important beta 1 or the ERB-2 in the wild type or in the mutant. So, and then that the mutant and the important beta 1 only showed up in the wild type with the ERB-2. And then looking at So by looking at the bottom panel, you can see that the ERB2 showed up in both the wild type and in the mutant. And this is just also, and it also just showed up in the ERBB-2 in both columns. So this just shows that the signaling pathways did work, but not when it came to the control MLGG. So one of the experiments that I proposed that they could have also done for this is the ELISA plate imaging experiment. And what this is an enzyme-linked immunosorbent assay. And it is a plate-based technique designed for detecting and quantifying peptides, proteins, antibodies, and hormones. This in an ELISA, an antigen must be immobilized to a solid surface and then complexed with an antibody that is linked to an enzyme. This could be a different way to find antigens in the experiment. Transport through the nuclear pore complex. So, um, the first bullet is talking about figure 4, which is the top figure on the right. And this is the transport through nuclear pore complex as a series of steps. And NLS containing cargo protein binds to important. The important Congo complex is then transported through the nuclear pore complex, which is the second step. The third step is nuclear RAN GTP binds to important, triggering the release of the cargo protein in the nucleus. The RAN GTP important complex is transported back to the cytosol. The hydrolysis of the GTP to GDP is accompanied by the release of important. So the next figure I'm going to talk about is figure 5, which is right below figure 4 on the right. And again, this was a third part to the experiment. So I'm going to try to explain this as best as I can. So by looking at the cytoplasmic fraction, you can see that the top panel in the ERBB-2 is present in all 3 of the levels. So you have your control, you have your wild type, and then you have your Q6NL mutant. So ERBB2 is a very great and show up. And then you have important beta 1 in the bottom level, which shows that, again, it shows up really well in the cytoplasmic and the control, the wild type, and the Q69L, because we're using the RAN Q69L thing. Then you look at the nuclear side, and you see that the ERBB2 shows up in all 3, but it doesn't show up quite as well in the Q69L. Then you go down to the bottom one, and you see that important B1 is faintly showing up in all 3. Then you have your total lysates figure, and you can see that the RAN does not show up at all in the control. It is not transfected in the cell at all, but the ERBB-2 is. So a mechanism of this is its clear translocation is unclear. The transmembrane receptors serve as sensors recognizing the growth factor in the extracellular environment and conveying the message through the signaling cascade to the nucleus. If we use RAN GGP NP instead of the RAN Q69L, it will have different results because the RAN GPP NP is a 9-hydrolysable form of RAN and will be localized in the nucleus. It binds to the important beta 1 in the ERBB-2 complex and causes the ERBB-2 to be released. So since it's localized in the nucleus, it won't leave the nucleus. So this will cause the important beta 1 and ERBB-2 to decline in the cytoplasm.